Okay, well, welcome. Today, unfortunately, we weren't able to make it into school. However, we will still be able to continue our investigation into the molecule of DNA. So, today, what we're going to really focus on is two things. First, we're going to focus on the structure of DNA. And the second, we're going to focus on how DNA replicates itself. So, we're going to look into the structure of DNA in some pretty good detail, and then just introduce how DNA replicates itself. So, let's review what we know up to this point. We know that there are two types of cells. We have prokaryotic cells, and we also have eukaryotic cells. We know that eukaryotic cells are cells that exist in our body. They're the cells that make up humans. Um, we know that eukaryotic cells actually store DNA inside of their nucleus. They have a distinct area that has that. Now, there's a second type of cell that's out there that's called a prokaryotic cell, and we know that a prokaryotic cell actually lacks a nucleus, and that's one of the big differences. We also know that eukaryotic cells tend to be part of a multicellular organism and can be very complex. We're going to focus on eukaryotic cells during this discussion, and leave prokaryotic cells for a future discussion. Now, last week we talked about the cell cycle, and we mentioned that DNA has to replicate itself within the cell cycle so that it can make enough copies so that when that cell divides, there's enough DNA to fill both of the cells. And we mentioned that DNA is copied during a phase called interphase. And we even kind of went into a little bit more detail and said that interphase is broken into three different sections, and DNA is actually copied in the S phase of interphase. So if you don't remember the cell cycle or you want to have a little bit of review, please look back at your notes regarding that because it's important that you understand the cell cycle before we move into DNA replication. So we also know that DNA gives us traits or physical characteristics that we see. So eye color, hair color, those different traits, those different characteristics that we have are coded within the molecule of DNA. So we're going to see how that is actually able to carry out those traits and those functions. We also know that traits are passed on from parent to offspring and that they're inherited through sections of the DNA called genes. So we are actually able to pass on those traits through these genes that we have. And all of this is basically coded within the structure of our DNA molecule. So DNA is pretty important to make sure that we can pass on these characteristics. Um, so that's kind of what we're going to look at, and we're going to see how the structure of the DNA allows it to be able. So first off, DNA is information. I mean, it controls everything that's about us. It controls the traits that we have, our blue eyes, our brown eyes. All of those characteristics are encoded in our molecule of DNA. DNA stands for deoxyribonucleic acid. It seems like it's a pretty difficult word. As we proceed through today, you'll actually be able to see where the different parts of that name comes from. DNA resembles a cookbook. Just like a cookbook contains all of the instructions to make a recipe, our DNA contains all of the instructions to make a person. And the way that DNA does that is by storing specific sections or segments that allow us to make proteins. And proteins are pretty important because proteins basically provide us with our traits. And if proteins aren't made correctly, then our traits aren't correct. And so that's kind of where we're at. And we're going to look at the way that DNA makes those proteins. So let's take a look here real quick. At this point in time, you should be able to kind of understand and remember what a trait is. You should be able to answer how or when, I'm sorry, DNA is copied. And you should also be able to describe what a gene is. So if you're not able to do that, then I would suggest pausing at this point in time. Go back to the beginning and just kind of review what we've been talking about, um, and you'll be able to see those things. All right, so if we're taking a look at what a trait is, remember we said a trait is a physical characteristic, something that we can see within an organism. So feathers or eye color, like we talked about, those are all distinguishing features or characteristics that they have. We also said that DNA is copied within the cell cycle, in the interphase part of the cell cycle, and then in the actual S phase of interphase. So it's copied during interphase of that cell cycle. And we also mentioned what a gene is. Now, a gene is kind of a small section of DNA that provides us with the coding or the sequence to make a protein. And we're going to look at that in more detail after we come back from break. But that specific DNA, that section of that DNA sequence provides us with those proteins. So we're going to look into that. So it's a little bit of an involved process, but we're going to take a look at that as we come back. All right, so let's really focus here first on the structure of DNA. I included kind of a rendition here up in the upper right-hand corner of what DNA looks like. And if you were to describe that molecule of DNA, a lot of people describe it as a twisted ladder. And if you would untwist that ladder, it would literally contain that molecule or that model that we're going to look at from there. The scientific name for the molecule of DNA is what they call a double helix. It's double helix because it's made out of two upright posts, which are twisted together, and then the rungs are part of those nitrogen bases, which we're going to kind of take a look at here as we move through. The molecule is made up of nucleotides, and if you look in the lower right-hand corner, we actually have the three components of what a nucleotide is made up of. So the very first thing that a nucleotide is made up of is a phosphate. The phosphate is connected to one of the carbons of the sugar, and in a DNA molecule, the sugar is deoxyribose. Now that's important to remember, so underline that in your notes, because deoxyribose helps us to tell the difference between this molecule and another molecule that we're going to be talking about down the road here. And the last part of our nucleotide is a base. Now, a base can vary. There are actually 4 different types of bases that are involved in the structure of DNA, and we'll take a look at those 4 different bases in general. But a nucleotide, highlight this, a nucleotide is made up of a sugar, a phosphate, and a base. So those are the 3 parts that make up a nucleotide, and it's important for us to remember those 3 parts. Now, we mentioned that the type of sugar within DNA is called deoxyribose sugar. Now, if you can take a look at it, in our molecule here, our molecule actually has our sugars, like we said earlier, in our upright part of our ladder structure. You can kind of visualize that ladder now. And the sugars are held together by our phosphates. So you can kind of see that nucleotide there. We have our sugar, phosphate, and then our bases. And the phosphates join together to the sugar molecules in a very strong bond. It's important that the sugar and the phosphate don't lose connection to one another because that's literally holding the molecule together. And so the DNA is formed by that sugar molecule and that phosphate molecule going up as we move up. And now also in this picture here, you can see that by the arrows indicating in our left side of our molecule that it runs in one direction, and our right side of our molecule actually runs in an opposite direction. And that's important to remember. DNA is referred to as being anti-parallel, which just means that one runs in one side and one runs in the opposite direction, which is important, but just as we know anti-parallel, that's what we're looking at, and that's what we're really focused on. So our phosphate group, a little bit more detail about that. Our phosphate groups bond with our sugars and they hold our nucleotides together and they form what they call a sugar phosphate backbone of our DNA strand. And it's important because that's what kind of provides that, like we said, structure for that DNA. You can also kind of see there are some bases, so that base T and that base A right at the top of our drawing there are nitrogen bases. Sometimes they're called nitrogenous bases. Nitrogen bases are fine for us. But you'll notice that T always bonds to A and G always bonds to C. And that's based on the shape of that molecule. And we'll kind of take a little bit more look at that here as we go through. So our nitrogen base. Basically, our nitrogen base are the building blocks for our DNA. So pretty much the DNA that we have, my DNA and your DNA, are very, very similar. And they're similar in the fact that the sequence of these letters of bases are pretty consistent. They only vary slightly within our DNA. And the sequence of these bases basically gives us what we call our genetic code. That genetic code provides those instructions to make those proteins that give us our traits. Let me repeat that. This genetic code provides the instructions that make proteins that give us our traits. The bases, like we looked at before, are actually connected to the sugar molecules of the nucleotide. And DNA has 4 distinct bases that you can kind of see. Now, we'll talk about these bases here a little bit, but take a look at the shape of these bases. The shape of these bases are referred to as being complementary. That's important. Highlight that. So complementary bases, if you look at the shape, they kind of resemble a puzzle piece. And A is only going to fit into T, and C is only going to fit into G. A and C, as you can see by their shape, will not ever connect together. And that's very, very important when we deal with DNA replication. And we'll talk a little bit about that when we get to DNA replication. But it's important that we understand those bases are complementary to one another. So they add together. So our 4 nitrogen bases that DNA has are adenine, which we'll remember by the letter A, thymine, which we'll remember by the letter T, guanine, which we'll remember by the letter G, and cytosine, which we'll remember by the letter C. So this picture actually shows how the molecule is formed. So our nitrogen bases are connected to our sugars, like we said previously. And you can kind of see the shape. Now, this is a little bit of a different shape. Obviously, this isn't the exact shape. It's just kind of how we're showing those shapes are complementary to one another. And you notice that A always bonds to T. Now, the way that they hold themselves together is through a hydrogen bond. Hydrogen bonds are weak bonds, and when we look at these hydrogen bonds, when we talk about them in our chemistry unit, we'll explain why they're a weak bond. But right now, just know that they are weak, and they actually hold our A and T together, as well as our C and our G together. So we'll see the importance of this hydrogen bond when we look at DNA replication. So, the structure of DNA, how was it discovered? Well, there was a couple different scientists that really played a vital role in it. I'm just going to introduce these scientists to you real quick so that you understand kind of the basis on them. There was an Austrian scientist by the name of Erwin Charkoff. He researched many different organisms in that, and he found that there was always an equal amount of adenine and an equal amount of thymine. And he conducted many different experiments and really concluded that. In fact, it came up, he actually has a rule named by him that's called Charkoff's rule, which means that A bonds to T and C always bonds to G. And we can look into more detail about that, but just know that that's kind of how scientists realized that A goes to T and C goes to G. So that's how we understood the nitrogen bases and how they connected to one another. But the molecule of DNA actually took a lot of other scientists that helped develop that molecule of DNA. And two British scientists, Rosalind Franklin and Maurice Wilkins, really were influential in coming up with the structure or the shape of that DNA. And they used X-ray photographs to be able to show that shape of the helix. Now, if you look in that lower right-hand corner, you can see that there's a central point and that it looks like things are kind of spiraled around. Now, it wasn't actually until Watson and Crick took a look at the discoveries that Rosalind Franklin and made, and they were the ones that were actually credited in developing the three-dimensional model of the DNA double helix. So Watson and Crick used the information, and you can kind of see how all scientists kind of work together to basically discover the unique aspects of the world of science. So it's pretty cool. So Watson and Crick came up with that molecule of DNA being a helix molecule. So basically, when we're looking at a double helix molecule, we see two strands of nucleotides, one running in one direction and one running in an opposite direction. We see that there's a sugar phosphate group bonded to one side, as well as the other side of that molecule has that same sugar phosphate group bonded going in the opposite directions. And because of Chargaff's rule, we know that A bonds to T and C bonds to G, and we have equal amounts of A and T and C and G together, held together by hydrogen bonds. So we have those complementary base pairs where A bonds to T and C bonds to G. So that's kind of the molecule, and when we get back, we're actually going to take a look into this structure. We're going to build a DNA molecule with kinetics and be able to kind of see this process and literally be able to manipulate it and see the double helix structure we have. So our final part of our process here is our replication of DNA. Now we're only going to talk a little bit, just briefly, about the replication process of our DNA. There's actually 3 steps in our replication process, and we'll try to make them as basic as possible. So our first is replication means that we're copying DNA. So we have DNA, it's copied, and it allows the molecule to form a brand new molecule. The second thing that we really want to kind of really focus on is the replication of DNA is referred to as semi-conservative. So DNA, semi-conservative, DNA means that there is one old strand of DNA, and you can kind of see it on our molecule here. So we have our old strand of DNA basically matched up to a brand new strand. So if you can kind of look at the bottom of that picture, the purpose and the reason why it's semi-conservative is because, remember, DNA codes for proteins, and it codes for proteins based on the sequence of these bases. So these bases need to keep steady, and they need to keep the same when we replicate that molecule of DNA. So that's the idea that we have there. It's called semi-conservative. So we take 2 strands that are separate, break them up with an enzyme. We then go ahead and we add nucleotides, which are basically floating around inside of the nucleus, and we add them onto that strand of that DNA. And then once they get added onto that strand of DNA, then throughout that process, we get them connected back together with our hydrogen bonds, and then it forms 2 brand new molecules of DNA. Like I said, this is a very quick overview of replication of DNA, but it is something that we're going to look into more detail with. But those 3 steps are the most important steps that we're going to talk about. So here's what I'm going to leave you with. When you come in tomorrow, these 3 questions should be answered along with the other activities that you are to complete with this. So our very first one is, what 3 components make up the nucleotide subunit? What are 4 bases, and what is the rule about how they pair together? And then finally, what are the 3 steps of DNA replication? So hopefully with this PowerPoint and with these notes, as well as with the other activities that you're going to do today, hopefully you'll have a pretty good understanding of how the DNA is made up into its particular structure, into its double helix, and also understand in basic detail how DNA replicates. So